Mature red blood cells are mainly composed of hemoglobin molecules, which give them the capacity to hold oxygen and deliver it to cells and tissues. But how are these molecules actually formed? It is important to take note that hemoglobin biosynthesis requires the presence of certain organelles for the process to be executed. Thus, the assembly and formation of hemoglobin occurs in immature red blood cells, which still contains organelles, beginning with the basophilic normoblast, also known as prorubicide, up until the reticulocyte stage only. Since mature red blood cells lacks the nucleus, mitochondria, and other organelles, hemoglobin synthesis no longer occurs here. Now from the term itself, we reckon the two main components of a hemoglobin molecule. Hemo, which pertains to the heme, and globin, which basically pertains to the globin or polypeptide chains. A single heme is formed from the incorporation of protoporphyrin IX and ferrous iron, while the globin chains are formed as a product of RNA transcription and translation. The formation, therefore, of a complete hemoglobin molecule first requires the synthesis of its two main components. First, we tackle heme biosynthesis, which occurs in both the mitochondria and the cytosol of immature red blood cells. Heme formation begins in the mitochondria. Here, glycine and succinyl-CoA condenses or combines to form an intermediate product termed alpha-amino-beta-ketoadipic acid, which undergoes decarboxylation catalyzed by the ALA synthase, leading to the formation of delta-aminolevulinic acid. This first step in heme biosynthesis actually requires the presence of pyridoxal or your vitamin B6. Two molecules of DALA is then brought towards the cytosol, where it undergoes condensation. The process is catalyzed by the enzyme ALA dehydratase, also known as porphobilinogen synthase, and results to the formation of porphobilinogen. Porphobilinogen exhibits a monopyral structure. Four molecules of the monopyral porphobilinogen then condenses, as catalyzed by the porphobilinogen deaminase enzyme, also known as methylbilane synthase, resulting to the formation of the tetrapyryl hydroxymethylbilane. In the presence of uroporphyrinogen 3 synthase, hydroxymethylbilane is then converted to uroporphyrinogen 3. It is to be noted, however, that there are actually two possibilities for hydroxymethylbilane. It is either converted to the uroporphyrinogen 3 isomer as stated, which is the one involved in normal heme formation, or it can be spontaneously converted to the uroporphyrinogen 1 isomer. What's important to understand here is the significance of the enzyme uroporphyrinogen 3 synthase. Generally, the absence of any enzyme related to heme synthesis results to a group of disorders called porphyrias. In case uroporphyrinogen 3 synthase is absent, normal heme synthesis cannot occur, and hydroxymethylbilane will be spontaneously converted to the uroporphyrinogen 1 isomer, which is not biologically useful. This isomer then accumulates as observed in a type of porphyria called congenital erythropoietic porphyria. Once uroporphyrinogen 3 is formed, it is then converted to coproporphyrinogen 3 as catalyzed by the uroporphyrinogen decarboxylase enzyme. This enzyme, however, does not target uroporphyrinogen 3 alone. In cases where uroporphyrinogen 1 isomer was previously formed, uroporphyrinogen decarboxylase may also act upon it and convert it to coproporphyrinogen 1. The succeeding enzyme coproporphyrinogen oxidase, however, is specific for coproporphyrinogen 3 only. Coproporphyrinogen 3 then moves into the mitochondrion, where it is acted upon by coproporphyrinogen oxidase, converting it to protoporphyrinogen 9. Protoporphyrinogen 9 is then converted to protoporphyrin 9 with the action of protoporphyrinogen oxidase enzyme. Lastly, ferrous iron is incorporated to protoporphyrin 9 with the enzyme ferrochelatase, leading to the formation of a heme molecule. Once heme concentration is adequate, this will send a signal to inhibit further synthesis so that excessive heme level can be avoided. The key rate-limiting mechanism is the inhibition of the synthesis and activity of ALA synthase. Once the enzyme is inhibited, the entire process will also be inhibited. regulated, maintaining heme concentration at the normal level. Also, as mentioned, deficiency of enzymes involved in heme biosynthesis is associated to porphyrias. Remember that for each enzyme deficiency, this corresponds to a particular type of porphyria. Now that we're done with heme synthesis, we proceed to the synthesis of the globin components. Globin synthesis occurs in the cytoplasm, specifically in the ribosomes. As proteins, the globin components of hemoglobin results from the RNA transcription and translation process. Each type of globin chain is coded by a corresponding structural gene, as seen here in the table. Remember that for most globin types, there are two corresponding genes present per chromosome, such as in beta, delta, epsilon, and zeta globin types. Whereas for both alpha and gamma globins, there are four corresponding genes that should be present per chromosome. During transcription, which occurs in the nucleus, the particular DNA segment holding the corresponding structural gene of the target globin type unwinds and then transcribed or copied, resulting to the formation of an mRNA. In this process, it is simply that the information from the DNA is copied into a format that can later be understood and translated by the ribosomes. mRNA then moves into the cytoplasm where it attaches itself to the ribosomes for translation. Here, it is deciphered and translated, resulting to the formation of a polypeptide chain, which elongates one amino acid at a time. This polypeptide chain contains the specific type, number, and arrangement of amino acids that corresponds to the globin type being synthesized. For most types of globin chains, there is a total of 146 amino acids, except for the alpha and zeta globins, which are only made up by 141 amino acids. The finished polypeptide chain will then be folded to assume their tertiary structure and are then made available in the cytoplasm, waiting for it to be assembled as part of a hemoglobin molecule. Depending on the hemoglobin variant needed, two types of globin chains are paired with each other. For instance, in adults, most of the red blood cells' hemoglobin will be of the hemoglobin A1 variant, and in such cases, the two types of globin chains to be paired are the alpha and beta globin. Once the main components are available, they are then assembled. Heme proceeds from the mitochondria into the cytosol, and each globin chain binds to a single heme molecule. Two dimers are then formed, with each dimer composed of two different types of globin that are bound by strong bonds, holding them into stable forms. In this illustration, the dimers are made up of one alpha globin and another non-alpha globin, which means that it could be beta, delta, gamma, zeta, or epsilon globin, depending on the variant that is to be formed. The two dimers then combine, forming a tetramer and exhibiting a quaternary structure. And this marks the completion of a hemoglobin molecule.